Hello students, now let's talk about the sixth part of the semi-conservative replication of DNA. Look, listen and learn. In this module, I'll be talking about the fifth step of the process, which is called as leading and lagging strands. Look at the names leading and lagging, and let us see why the strands are called as leading and lagging strands. Look at this picture of the separated DNA strands, okay? Check their orientations, right? What were these strands called as? Template strands. First, I chose the left strand as the template strand. Yes? Also recollect that template strand has to be oriented from 3' to 5' direction, right? What happened at the 3' end? At the 3' end, primer was bound. Yes? And with the help of DNA polymerase, what did it do? It basically synthesized the new strand continuously, right? From 5' to 3' direction. Now this template strand, I'm going to give it a name and I'm going to call it as the leading template. Is it okay? Because it leads in the process, right? Then this new strand that was built on the leading template will be called as the leading strand, as you can see here. Fine. Now look at the opposite strand, okay? Guess at this strand if this was the template strand, right? Check its orientation. Now this strand is going to be from 5' to 3' in the same direction, which means the replication will begin from the other end, correct? Fine. Now there is a primer. Yes, it will always be attached to the 3' end. You know that too. What will happen next? You guessed it right. DNA polymerase enzyme. Where will it bind? Next to the primer. What would it do? It will scan the template and add the complementary bases, right? And then the strand will be synthesized from 5' to 3'. But you see, only as a short fragment. We still have to complete the process. How would we do that? Once again, we'll have a primer. Once again, there'll be polymerase. And once again, the polymerase will scan the template and add the complementary bases in the new strand. But again, only in a small fragment from 5' to 3'. Is that okay? Still, replication is remaining. Now what will happen? Once again, you will see the primer. Once again, there will be DNA polymerase. And once again, it will scan the template and keep adding the complementary base pairs in the new strand from the 5' to the 3' end. You guessed it right. You see that there are only small fragments, but you see how long it took? It took a while, right? And therefore, this template is called as the lagging template. And this new strand is called as the lagging strand. Although it is made simultaneously, but because it takes more time, it is called as the lagging template, and the new strand, which is synthesized on it, is called as the lagging strand. Okay? Now remember, these fragments that you see, they have a name, right? And these strands are simply called as Okazaki fragments because it was discovered by a Japanese scientist named Reiji Okazaki. He was the one who actually decided that this is how the lagging template will be synthesized. Is that okay? Now, look at the lagging strand. What do you see? You see there are primers still remaining. Look at the leading strand. There's one primer right there too. The primers are RNA in nature, so they have to be removed. Is that okay? To remove the RNA, we require another enzyme, and this enzyme is called as RNase enzyme. What will it do? The RNase enzyme will bind to the primers. One there, one there, one there, and one on the leading strand. And what will it do? It will simply remove the primers. Now look at the lagging strand. See there are empty spaces, but you can still see the Okazaki fragments. So the empty spaces and the Okazaki fragments have to be joined so that the lagging strand is also complete. And who will do that? Yet another enzyme called as DNA ligase enzyme. What will it do? It will start up there at the 3' end of the lagging template and will simply complete the two strand. Is that okay? And it will do so once again from the 5' to the 3' direction. Is that okay? And this strand was your lagging strand. Fine. The leading strand was synthesized continuously, so it is also called as the continuous strand, while the lagging strand was synthesized discontinuously in the form of short fragments, so it is also called as the discontinuous strand. Is that okay? Going back to their original names, and this is the concept of the leading and the lagging new strands. Is that okay? This brings us to the end of 6th part of the semi-conservative replication of DNA. In the next module, I'll be talking about the 7th part of the semi-conservative replication of DNA. I will also throw light on the process of replication in prokaryotes. Thank you and take care.